At the end of this molecular evolution course, we're going to look at two major classes of protein evolution, but we're also, for both of them, going to give you a little bit of morphological and historical background. So this unit is going to concentrate on the first of those, which is looking at opsin, a class of G protein-coupled receptors, and the tissues in which they're expressed. So opsins are very tightly and closely associated with eyes, with the organs that allow us to form visions, and also are found commonly across the animal kingdom. So we want to talk a little bit about eyes before we get into the molecular side. So as we look across the tree of life, it's clearly apparent that the origin of eyes is not a very simple event in the history of animals. So there are three major types of eyes when we're going to talk about fully forming images. And then there are, in other metazoan groups, metazoa is the technical term for what is also sometimes called animalia, so among animals, there are other groups of photoreceptors of various types, but it's obvious that the eye as a complex organ evolved at least three separate times. And in fact, originally, all three of these origins of eyes were thought to be separate and completely independent. So they were not thought to be homologous, and there was thought to be convergence among them. So let's classify these. You should know these three major image-forming type of eyes. It's more complex if we just get into just photoreceptors, and although we'll mention them in a couple groups, we won't study or learn them in detail like we will with the complex eye. So complex image-forming organs, or eyes if we want to call them that, are found in three major groups of organisms. We find them among the arthropods, which are characterized by these complex, multifaceted, often called a compound eye. And so in the arthropod eye, each individual rhabdomere, as they're called, you don't need to know that name, but each facet acts as something of an independent unit, but then they are all integrated together to form the final signal that is then interpreted by the brain. And they can see many colors, in fact, probably a wider range in most arthropods than in any of the other eyes that we'll talk about here. So there's a wide range. And there also appears to be regional specialization, so that some parts of the eyes might be dedicated to seeing some wavelengths of light, and other parts of the eye may be dedicated to seeing other wavelengths of light. And that's a complex image or complex picture across the diversity of arthropods. And we'll mention that briefly here, but in a little bit. But now just know the arthropod eye as one of the main examples of the origin of eyes. The other two include the vertebrate eye, which of course this is a diagram of the human eye. The human eye is just one example. Almost all vertebrates have eyes, or at least remnants of ancestral eyes, right? It's a thing that is unique to all modern vertebrates and appears to evolve fairly early on in the history of the chordates and became a defining feature, one of the defining features of the vertebrates. So these are complex structures, of course, integrating nervous tissue and connective tissue and muscles and all sorts of other things. In the human eye, we have a lens that the shape is changed via these muscles, these ciliary body muscles that pull on either end of the lens and change the shape so that we can focus an image back here on the retina. There's a single origin here on the back of the retina that then goes back to form the optic nerve. That's also a blind spot because there's no sensory tissue there. Okay, now the third type that you should be aware of, and we're going to compare and contrast this with the vertebrate eye because it is much more similar to the vertebrate eye than it is to the arthropod eye, which is really unique among eyes. But despite the overall similarities, at least some similarities between the mollusk eye, as represented here in the octopus, and the vertebrate eye, as represented here in the human, there are clear, distinct differences. And this is one of the reasons why originally these were thought to be completely independent origins of the eye. We'll talk about that here and define deep homology in just a little bit. So the octopus also has a refractive lens. However, it is held relatively static. It does not move and cannot change shape like the vertebrate eye. So for an octopus to function its eye, the length of the eye is changed, and that focuses the image at different distances. At rest, it can focus in the distance, and then if it needs to see close up, it can be the position of the lens can be changed. But notice there's also an iris, right? So there's also a way to change the amount of light that's coming in. There's a cornea for protection. Notice that the retina, although superficially similar, in the deeper structure is actually quite different. There's no single optic nerve. And also a major difference here is the pigments, the light sensing pigments, these opsins that we'll define here in a little bit. In the vertebrate eye, they're pointing back. So it's almost like a little bit of reverse. But in the octopus eye, they're pointing forward, and then all of the nerves then just go right back, and then eventually they will coalesce to a single one, but there's not a single optic nerve that comes out like here. And so you can see some superficial differences, but also these distinct differences in the structure of the retina, the way the image is formed, and several other things that we won't mention. Those all gave rise to the idea, and phylogenetically it is supported because the octopus is part of the mollusks, a very distantly related group from the vertebrates, and there are groups in between, phylogenetically intermediate in here, that are not, don't have a complex eye. Yet, as we begin to examine the origin of these eyes developmentally, we notice some very deep similarities developmentally that we won't get into the details of. So there appears to be a developmental connection in the genes that are causing these eyes to grow. And so the idea is, and this has been borne out both with genetic studies and now with fossil studies and mapping of all of the eyes onto the origin of the metazoa, is that the eye, although it did evolve as a complex structure independently 3 times, the eye as a photoreceptor, and so maybe we wouldn't call it an eye at that point. So rather a photoreceptor as a light-sensing organ is something that is homologous to all animals, although it's been lost in some lineages. But there's this independent single origin of a photoreceptor at the base of metazoa, maybe even in the earliest metazoa, or at least very early in the history of the metazoa. There's still some uncertainty of that. And so all of these eyes that we see, the arthropod eye, the vertebrate eye, the mollusk eye, and then the other light-sensing organs across other groups are all homologous, although they have been elaborated and differentiated into the various complex eyes separately. So to be very, very kind of precise and clear about this, the photoreceptor that is common to all animals and has evolved into complex eye 3 separate times, that photoreceptor is homologous. However, the complex eye itself and the way it forms images in the arthropod, the vertebrate, and the mollusk is analogous. And so we see some elements of convergence, particularly between the vertebrate eye and the mollusk eye, but it is a separate independent event as far as becoming a fully forming image eye. Okay, so just be aware of that. And so there's this amazing amount of diversity that we can look at that we won't get into really in much detail. We'll hit a couple kind of highlights here, but just in the number of photoreceptors, the way the photoreceptors are arranged with these clear, distinct regions this is a mantis shrimp eye. These are all examples of arthropod eyes. So even within the arthropod eye, there's this huge, amazing range. And we, as we are beginning to look, are finding as much diversity and complexity at the genetic level as you might expect. Okay, so just be aware that this is a great model for morphological diversity and also a model for genetic diversity. And we'll talk, we won't really get into the morphological diversity among arthropods. It's a cool subject, but beyond the scope of what we want to do for this course. But we will look a little bit at the molecular diversity. So one other last very interesting thing about morphology before we move on to the molecular diversity of this group of proteins is that the mollusk eyes actually serve as a really great example of how we can get a complex integrated structure with muscles and connective tissue and lenses and neurological tissue and all these other things that work in concert, how we can get all of that complexity and how it can evolve stepwise from a very, very simple origin. Now, we don't have this in vertebrates because all of the intermediate lineages in vertebrates are only represented in the fossil record, and they're very limited because this is fairly soft tissue and very rarely is preserved. We'll look at one example here a little bit later on, just as an aside. But amongst the mollusks, various groups have maintained many of these independent steps along the way to developing a fully formed eye, like we see in the octopus, squid, nautilus. To some extent, even the nautilus, which is kind of cool, has an intermediate one. So it's really in the octopus and the squid that we see this fully formed eye. Okay, so amongst the different types of mollusks, we have everything from simple photoreceptors, which are probably somewhat similar to the ancestor in all metazoa. We have some that form this little cup-like shape that allow for perhaps direction, or maybe a little bit better about intensity of light. And then we have a nautilus eye, which is like a pinhole camera that can form a very simple image but can't focus close up and is so limited to its shape. And again, then in murex, which are some snails, we have a little bit more of a complex eye, which seems to have evolved independently in at least having a lens, although there's no way to focus this lens, and again, is probably forming a very, very rough image. There are even some very cool ones which aren't listed here among some types of mollusks that use iron pyrites and other things to form these very rudimentary coverings and very, very tough coverings over the eye. So it's a lot of cool diversity that we don't really have time to get into, but we will maybe look at a little bit later on for the graduate course. Okay, and then finally, of course, the complex octopus eye, which is a fully image-forming eye. In some ways, a little bit even better designed, although that I hesitate to use that term because, of course, these evolved and were not designed ahead of time. And we can see some of the limitations of that in the vertebrate eye with the blind spot, with the subject of the lens. The lens eventually loses its elasticity, and so when you get to be 45, 50 in humans, you have to wear reading glasses because the lens is no longer elastic. But in octopuses, that's not a problem, right? Technically, so just to be clear, the technical plural for octopuses should be octopoda, right? Because that's the pluralization of that Greek root word, but that always sounds silly. So octopuses, if someone says octopi, you know that they are trying to be smarter than they really are, because octopi would be how you would pluralize a Latin root, but poda is a Greek root, so it shouldn't be take a Latin pluralization. Anyway, that's an aside. I'm not going to ask you to give me the correct pluralization of octopus, but technically it's octopuses, or if you wanted to be really stuck up, octopoda. But anyway, octopuses is plural. So in the octopuses, we have this really well-designed eye. Octopuses would never need glasses, even if they could wear them, which would be hard since they don't have a nose. But they would never need glasses because they focus very, very differently than the vertebrate eye does. So just an example of some of the differences and some of the different pathways going to a somewhat similar outcome, but with different steps along the way. All right, so let's now delve into the molecular diversity that goes along with the eye. The gene that is most closely associated with the eye, and in fact is responsible for light sensing in the ancestral form, and for image formation in our more derived forms, is a group of proteins. We'll call it a family of proteins called the opsins. So this is an opsin gene family. They are readily identifiable by a number of different domains that are similar across the opsins, but the most easily identifiable set of domains are these 7 transmembrane domains, which are diagrammed here in a linear form, but here in this tape and ribbon and tape form, you can see kind of how these 1, 2, 3, 4, 5, the purple ones there in the back, 6 and 7 transmembrane domains are arranged. And then we see it in various other forms in a space-filling model over here with some indication by color onto what parts of this gene are more from the front side and the back side are more constrained and which ones are less constrained. Okay, but anyway, they are part of the G protein coupled receptor, GPCR. It's a huge, huge superfamily that does many other things other than light sensing. But the opsins seem to have evolved very early on in the history of the metazoa for sensing light. However, there's still some debate about that. They may have had another purpose. Well, certainly as a G-coupled protein, they had another purpose earlier for sensing light and only more recently gained that. But even among just what we now identify and call the opsins, there's some debate as to whether their original function was light sensing or that have taken that on secondarily. And so we'll look at that here a little bit on a phylogeny. Okay, but within this superfamily, within this family, which is part of the superfamily of these G protein coupled receptors, we have 4 main groups. And so now we're going to be looking at the material that you've hopefully reviewed, at least gone over a little bit in that Porter et al. Paper. And we're going to give you a summary of what we know and look at both kind of how phylogenies help to us inform about the evolution of this group of families and a little bit of some of the methodologies that we've talked about now applied to this family of proteins. So there are 4 main distinguishable groups across the opsins. Here, they're color-coded. We have these group 4 opsins, which again, they've subdivided into 4 subgroups, but group 4 opsins. We have the C-type opsins, which are the major visual opsins of vertebrates. So when we're talking about human vision, we're talking about a C-type opsin. And then these R-types opsins in blue and green shades here, which are the major visual opsins for the arthropods. So not only do arthropods have a distinct morphological but fully functional eye, they also are molecularly quite different because they primarily use these as their visual opsins. The group 4 opsins, by the way, are very divergent in function. Many of them are expressed only in the brain, and so probably, even if they did ancestrally, no longer have a light sensing function, although there's some interesting connections with some of them and the circadian rhythm. So although they may not sense light directly, they are still connected at some level and play roles in the day to night functioning of the body and how that is regulated. And then finally, this 4th type, which is unique to the Cnidaria, thus the name Cnidops. So this demonstrates that this group probably evolved very, very early on and then has diversified differently in different groups of animal. So although the jellyfish, right, and other Cnidaria don't have light sensing organs, they do appear to have light sensing capability that is more diffuse and not in one single group, at least among most of the Cnidaria. So although the vast majority of jellyfish and ctenophores, which are the comb jellies, don't have any visual structures at all that sense light, there are very few that appear to have evolved light photoreceptors, not fully formed eyes, but photoreceptors separately. So I've got an image here of a box jellyfish, and these little dark areas that are clusters of a few dozen cells that allow it to sense light. But many other Cnidaria can still sense light, but they just don't have clusters of cells. It's done at a single cellular level. So a much simpler way in it they've evolved seems to be somewhat simpler and have not evolved to the extent that the eye has in the mollusks and the arthropods and the vertebra, so among the higher metazoan. So it's very clear and obvious just from a phylogeny here, and if we look in the subsequent one, we'll look at distribution of different model organisms and their opsins, that there have been many, many, many gene duplications and many gene losses in this family. So it's a really, really complicated story, particularly if we try to do the overall whole story. And in some cases, it's more informative to look at smaller subgroups and look at what's going on there. Although even there's some really amazing examples of rapid duplication, gene duplication, and subsequent gene loss. So among these 4 major main groups, we see all sorts of instances of convergence in function and even convergence in location of where they are expressed. So it's a very labile and evolutionarily it seems to change quite easily, and there have been lots of different options that have explored. Also, amongst these opsins, there has been a lot of change in evolution at the 3rd position of these protein coding areas, and so we often have high saturation followed by constraint at other areas, and so they can be really tricky because that's a tricky thing, and that happens also in a lot of mitochondrial genes where we have 3rd positions have evolved so much that they're saturated and no longer useful for phylogenetic analysis, and then 2nd positions that are so conserved that there's just not a lot of data there. And so it can be a little bit tricky, in the analysis of these phylogenetically was not just as simple and straightforward as many other phylogenetic analyses. So an added additional stumble to this is that there are no really close G-coupled proteins that are the obvious sister group to all of the opsins. So it's obvious we use G-coupled proteins because they're part of that superfamily, but which one? And so in this analysis, they used a couple dozen genes as outgroups to help give it a better rooting, but many of these seem to have evolved and are quite different, and so alignment and analysis can be very difficult in this group. But it's an exciting group to study. And so one way that they've been able to kind of get around this and see some patterns is, instead of relying on the DNA sequence, they can rely on introns and intron arrangements or other features of the gene that in some cases are a little bit more conserved. And so by doing that, they're able to get quite a nice picture of what's going on, but still with a lot of work to do. So notice that the fish, as represented by Daniel Ririo, these little fish icons represent an opsin that's been identified from the fish. So fish have this amazing diversity of opsins, and it's still not been characterized whether all of them are visual. And then we have the human one, which is here and here, a couple that are closely related there, and another one there. And this represents the three visual opsins in humans that are for color, and the one visual opsin that is for just sensing dark versus light. You can see mouse, and then here these little fruit flies, and that's the horseshoe crab, which is more closely related to spiders and insects. But you can see the diversity of these genes up here. These are primarily the arthropod genes in the R type. Although some of the R type, notice, are found in humans and fish, they're just not visual. They have other functions, some of which are characterized and some of which aren't. So again, it's a very cool group, but very tricky, and there's no overall single pattern, although we do see some things emerging, like the visual difference where arthropods rely on the R opsin, and humans and other vertebrates rely primarily on these C-type opsins. Okay? And so I just want to go one more thing kind of back in history, that many of these opsins no longer have visual function. They're expressed only in the brain. Like, for instance, pinopsin, which is now largely in the brain. It was originally, the name, as the name implies, was isolated from the pineal gland in the brain. So it's an area where there really is no light to sense. So it's still doing signal transduction, but doing something else. And there's some debate as to whether this was an ancestral one that has now taken on a brain function. This, by the way, is an image of a very early, well-preserved placoderm eye, which shows you some of the intermediate morphological stages that might have corresponded with pinopsin. And there's some argument that pinopsin may have been the original just light-sensing eye gene, and then other parts of the family duplicated off and took on the main function of light sensing, and pinopsin now is regulated to expression in the brain for other functions. So again, it's not an easy story. And in fact, at the end of this paper, they've got some diagrams. There's several of them. I just picked one where they basically show this. So this is the same phylogeny. We've got the R-type, C-type, Nidop. So this is the same phylogeny, but rather than color-coding it based on the types that they have identified, they are trying to show you where the genes are expressed. So blue is expression in the eye. And so notice that eye expression is common across all types of these. And they're using the term eye loosely, any sort of photoreceptor. So here are the cubozoa ones that we talked about in the Cnidaria. Some of them are expressed only in the brain, the ones that are red. Others are expressed in the eye and the brain. Some rare ones are expressed in the skin. This is particularly, I think, true amongst some of the mollusks where they have photosensitivity in the skin. And then there's some that are kind of oddballs and expressed in other tissues also. So the lines that they've got color-coded here, they're trying to look at the type of cells. And the take-home message to this, that there is no clear pattern. We don't have a group of genes that are only in eyes and then a group that are only in the brain. So there's lots of flexibility, and have been multiple gene duplications and co-option of location to take on a new, or at least a secondary and sometimes then completely new location. And that tissue types, there's no clear correlation between tissue types, although some general rules do apply. So ciliary bodies are primarily the C-type opsins, which is where the name comes from, but there are some exceptions to the rule. Notice the green and the pink ones, which represents expressions in the neurons or rhabdomeric regions. And then these are primarily expressed in the rhabdomeric, which are those individual facets of the arthropod eye, but again, with some exceptions to the rule. So I'm just going to end with this quote from the Porter et al. Paper, which kind of sums up the molecular evolution. And then I'll look at one last kind of fun ongoing piece of work that we won't look at a single paper for, but is an interesting group. So near the end of the Porter et al. Paper, they say the evolutionary history presented here illustrates the lability, good vocabulary word, that means flexibility, basically, ability to change, evolve, and mutate. But the lability of the opsin protein in diverse photodetection systems to couple to new G protein pathways. So it's also, I didn't put that image here, but also the other pathways that they interact with is very diverse and doesn't show a clear, distinct pattern either. And to be promiscuously expressed in a wide range of cell and tissue types, which is illustrated here in this diagram. And so finally, to completely finish this, let's just look at an example of extreme, both at the morphological level and at the molecular level. Arthropods are very, very cool because they have such a diverse range in number of eyes. Some of them have photoreceptors or multiple fully image-forming eyes. This little tiny water flea, which has fairly simple eyes, has more than 30 opsins, and many of which are uncharacterized. They don't know if they all serve a photoreceptor purpose. I think the mantis shrimp, which most of them have been identified as a photoreceptive purpose, has as many as 17 opsins. So there's some discussion. They can see far, far into the UV range, which humans can't, into the infrared range also. So they have a much wider visual spectrum than humans, and they can probably make much more fine distinctions between different shades of colors. And so really, really a complex and in some ways more advanced visual system than any vertebrate eye. So it's a very cool but complex group to study, and amongst the arthropods is especially complex and diverse. Dragonflies have somewhere between, depending on the species, between a dozen to 2 or 3 dozen different opsins, some of which are not even characterized at all.